Live, sir.
Good evening. It's great that all of you are here. I am Neeraj Jain. I am currently Director of National Brain Research Center. You can look it up on your internet anytime you want to know more about this Brain Research Center. And I'm glad to be chairing today's session, which is the first in the series of INSA Foreign Fellows. INSA is Indian National Science Academy, which is one of the 3 science academies of India. And the greatest thing is Professor Mriganka Soor is also with us, who will be giving this talk. With me, I have 3 people who are helping in the lecture, who are panelists on this. One is Professor Vidita Vaidya. She is professor at Tata Institute of Fundamental Research and Department of Biological Sciences in Mumbai. I have Arun Shripati, who is professor at Center for Neuroscience in Indian Institute of Science, Bangalore. And Professor Subrat Sinha. He is head of the department and professor at biochemistry at All India Institute of Medical Sciences in New Delhi. Before we introduce the speaker, Professor Mriganka Soor, I'll introduce Dr. Chandrima Shah. She is current president of INSA and is a very active person who is introducing a lot of new activities, including this lecture series. Professor Chandrima, I would like you to tell us something about this lecture series. Yeah, please go ahead.
Yes, thank you, Neeraj. So all distinguished guests and viewers of for today's program, on behalf of INSA, I extend a very warm welcome to all. We are starting this series of lectures by the Foreign Fellows of INSA to get a feel about the kind of work that our fellows from other parts of the globe are involved with. This will be a series of distinguished lectures in sciences. We already have a running series of lectures of eminent scientists from the country. I would like to thank the Science and Engineering Research Board for generous funding. We are delighted today that Professor Mriganka Soor could join us as the first speaker of the series. His hugely important work generates much curiosity among scientists as well as the general public. It is a delight to have him with us today. For a bit of history, the Academy has been inducting foreign fellows since 1935, but till 1973, they were known as honorary fellows. Presently, there are 94 foreign fellows of INSA, with 5 recommended for next year as well. So the total tally will go to 99 next year. All very distinguished scientists, and 11 of them are Nobel laureates. Maximum number of fellows are from the United Kingdom and the United States of America, and also Germany having a reasonable share. We expect that this series will promote interactions of foreign fellows with our young scientists, and fellows with foreign counterparts will spark debates about science. And we hope that this will enthuse our young scientists of the country. It would be an ideal situation when we should be able to meet in person. But till then, this is the best way, and we hope that this series will be enjoyable to all. Thank you. I ask Neeraj, Dr. Neeraj, to do the further proceedings.
Thank you, Dr. Chandrima. Thank you for that introduction. And I see we have a lot of audience today. The number is increasing and chat is increasing. And once again, good evening, good afternoon, good morning to all of you, depending upon where you are on the globe. Before I introduce Professor Soor, I would like to tell you that you can post your questions in the chat on YouTube. We'll be monitoring that. That will be at the end of the lecture, and we'll pick questions from that. and ask them on behalf of you to Professor Sur, who will answer those questions. Professor Sur, if you do neuroscience anywhere in the world, you know him. He's done phenomenal work. It doesn't need too much of an introduction. His interests span from looking at neurons at down to synapse level to entire systems level. I have known him for a very long time. Phenomenal, very interesting work, irrespective of whatever question he has picked up. He has done top of the line work always. He is at MIT and also in McGowan Center there. He runs the Simon Center at MIT, and he has been the head of the department there for a very long time, a chair that he has very recently given up. Since this is an online series, I will not have a very long introduction because you can all look at the website for him. His details have been already circulated. So without much delay, I like to invite Professor Mriganka Sur to start his lecture. Professor Sur.
Thank you very much, Neeraj. Let me share my screen and begin my slides. All right. Well, I'm very honored that the Indian National Science Academy asked me to speak in their Foreign Fellows lecture series. It is a great honor to be elected a member of the Indian National Science Academy, and particularly if you are from India. I'm also acutely aware of the conditions under which we are having this series and my talk under a global pandemic, and particularly our respective countries. I now live and work in the United States, and the United States has not done well in its ability to control COVID, and I hope India does better too. But the only hope of getting COVID under control is through science and through a scientific attitude and understanding of infectious diseases and how we might deal with them. So today I'm going to talk about a broad swath of work that my laboratory has done, and I'll place it in the context of what I think is perhaps one of the most important issues and questions of our time, which is, how does the brain create the mind, or the neural architecture of cognition? And so we'll start with the fact that the human brain has nearly 80 billion, with a B, neurons, an equal number of non-neuronal cells called glia. And the concerted activity of these cells creates the mind. And of course, these brain cells are not organized randomly in the brain. There are different structures and modules and information processing regions that interconnect with each other and within each other that give rise to cognition. Some definitions. Cognition, as defined in the Oxford English Dictionary, is the mental action or process of acquiring knowledge and understanding through thought, through experience, and the senses. That's a pretty broad definition. And commonly, cognition refers to all activities of the mind that underlies perceiving, understanding, learning, memory, attention, communication, such as language or other forms of communication, and even reasoning. And so how might we think about how does the brain give rise to cognition? And let's start with a simple example, or not so simple, but it's illustrative. Here are 4 self-portraits by a German artist called Anton Rederscheid, who died many years ago. But some years before his death, Rederscheid had a stroke. And he had a stroke in a particular part of his cerebral cortex. A stroke is a situation where blood vessels are either blocked or they burst, or they hemorrhage, and therefore a part of the brain is deprived of oxygen and hence ceases to function. And Rederscheid, after his stroke, looks in a mirror and he draws self-portraits. And these are 4 self-portraits. I hope you can see my mouse. 4 self-portraits done soon after the stroke, a few months later, a few months later, and a few months later still. And a trained neurologist can take one look at these images and tell you where his stroke was, which is a remarkable example of our understanding of brain function, but also how far we need to go. So Rederscheid takes a look at the mirror and he draws half a face. The left side of his drawing is completely blank. He draws one eye, half a nose, and potentially half a mouth, and one ear. The left side is blank. It is not that he cannot see. There is nothing wrong with his eyes. There is something deeply wrong with his ability to integrate information from his eyes, and importantly, to be aware that there is the left side of his face that also needs to be drawn. And then a few more months, and the left side of Rederscheid's self-portrait is still impoverished compared to the right side. And a few more months, and Rederscheid realizes there are other parts of the brain other than the location of the stroke that also contribute to cognition. And he turns his face away, and then a few more months, and Rederscheid was lucky. Usually when brain cells die, particularly if they are deprived of oxygen for a long time, they don't recover. But then occasionally they do, and often, if the stroke area is small, other regions around the infarct take up lost functions. And that's likely what happened in Rederscheid's case. So where was Rederscheid's stroke? Rederscheid's stroke was in a part of the brain called the right parietal cortex, right here in the middle of my brain, but on my right side. And if that happens to me, then I am unaware. I do not attend to things on the left side of the world, because each side of the world projects into the opposite side of the brain and is represented there. So then, this tells us at a glance that cognition has different components. One of them is attention, another is visual perception, and it arises from an internal representation of the world. There is nothing wrong with Rederscheid's face or with his ability to see, but there is something deeply wrong with the way that he is internally representing this external world, or at least the world of his face as seen in the mirror. And this representation involves different brain regions and the dynamic activity of these regions. In this case, it tells us that the parietal cortex is a critical node in integrating vision and even attention such that one can perceive what exists. So that leads to the proposal that analyzing neuronal activity across brain regions, not only in one brain region, but other regions that feed into critical nodes, such as the right parietal cortex, understanding the pathways and circuits into these regions is crucial for understanding brain functions underlying cognition. A little bit of basic neurobiology. I told you the brain is made of neurons and also non-neuronal cells, but neurons are unique cell types. They share many things in common with cells, with the trillions and trillions of cells that make up the rest of our body, but they have 2 unique features. Neurons encode and transmit information via electrical impulses. because they have a semi-permeable membrane that allows them to fire action potential. Sodium comes in, potassium goes out, and that leads to the action potential, which you can record from within neurons. And these are these all or none digital pulses. There is no information in the height of the pulses or the electrical spikes. All of the information is in the interval between electrical spikes. And we will keep coming back to this as we talk about information processing in the brain. What do I mean? I mean the way that neurons fire action potentials and how they modulate the interval between spikes in order to convey information. It's a remarkable thing. Everything we do, the fact that you can see me on your screen, is based fundamentally on the information contained in the spikes that your eye and brain and the rest of your mind are constructing from your senses. The second thing that makes neurons special is that they make synapses. A neuron has a beginning, a wire, and an end, and it physically ends before it makes contact with the next neuron. But at that ending, there is a specialized junction called the synapse, where the electrical impulse has to stop because there is nowhere for it to go. There is a gap between neuron 1 and neuron 2. But that electrical impulse leads to the release of chemical transmitter from the presynaptic neuron, which is taken up by the postsynaptic neuron through neurotransmitter receptors. The most important of the neurotransmitters in the brain being glutamate, that leads to excitatory transmission. But there are inhibitory transmitters like GABA that lead to a reduction in the propensity of the postsynaptic cell to fire. So glutamate leads to increase in the propensity of the postsynaptic cell to fire. It leads to excitatory transmission. GABA leads to inhibitory transmission. And together, neuron 1 influences neuron 2 or 3 or 4. The average neuron in the brain connects to hundreds of other neurons via thousands of synapses. And this is the idea that neurons connect to form networks. And the organization of neurons via synapses into networks is at the heart of how computations in the brain come about. 100 neurons wired together via synapses that themselves modulate incoming information. It is not the case that every spike in the presynaptic neuron leads to a spike in the postsynaptic neuron. You need integration, you need summation, you need inhibition in order to carry out computations that are the property, they are the engine of the brain.
Professor Soor, if I may interrupt briefly, INSA wants you to come slightly closer to your laptop, if possible at all.
Okay, is that because you can't hear me?
Yeah, because
okay.
the light is hearing you as well, so that's why.
Okay, can you hear me now? Is that better?
Yeah.
I should just speak louder.
Yeah.
All
yes.
right.
That's better. Yeah. Okay.
That's better. All right.
Thank you. Sorry to interrupt.
No problem. I hope what I said was understandable, because it would be a shame if I have to repeat all of that. Okay, so new technologies are transforming our understanding of brain function and structure and processing, and these technologies span many different levels of scale. Most of you are aware of brain imaging via functional magnetic resonance imaging or positron emission tomography. This is whole brain technologies that lead to understanding the brain on the scale of millimeters, but then at least in animals, we can remove the skull and peer deeper and deeper into the brain. We can use optical tools to understand population activity on the level of thousands of neurons via optical methods. We can put electrodes in the brain to understand the activity of single neurons. We can go still deeper using other optical tools such as multiphoton imaging to look at the activity of individual neurons and molecules within neurons. Here is the postsynaptic element of a neuron called a spine, and now we are talking on the order of microns. I remind you that a micron is 1/100th the width of a human hair. And then even newer technologies enable us to look at the molecules within single synapses or single neurons at the level of submicron resolution using super resolution methods. So all of these technologies give us unprecedented windows into the brain, and they allow us to ask interesting, deep questions, such as how does brain activity, potentially spanning all these levels of analysis, give rise to brain functions up to and including our highest brain functions. So today we have two questions. How is the brain wired? And I'll speak to that very briefly, but understanding how the brain wired allows us to understand at least a little bit, even in the space of 40 minutes or so, how does the brain change? And understanding how the brain changes is crucial to understanding how does brain wiring create cognition. So how is the brain wired? Brain wiring may be studied in different brain regions for different processes. And one way that my lab has studied it is to understand the processing of vision. So the visual system begins with the eyes, but is created in the brain. I already showed you Anton Rederscheid's self-portraits, where even though there is nothing wrong with his eyes, a stroke in the brain affects his ability to see, at least construct, the visual world on the side opposite to his stroke. So visual cognition arises from information sent by pixels or retinal ganglion cells, but that go through the optic nerve to the primary visual cortex, and there a hierarchical distributed network of areas down various serial and parallel processing pathways gives rise to vision. Later in my talk, I'll pick up on these regions, the parietal cortex, the frontal cortex, and we'll discuss how information from the primary visual cortex gets transformed as it goes into parietal and frontal cortex, at least in animals, such as the humble mouse. But the mouse shares in common with primates, including humans, the basic fundamental architecture of the brain that allows not only visual processing to happen, but many different kinds of cognition. So coming back to vision, a Nobel Prize was given to Hubel and Wiesel now some 40 years ago for making a very profound discovery. It's the simplest of discoveries, which is that cells in the primary visual cortex, or V1, are responsive not to pixels or dots or blobs of light, but to a very particular organization of light. And that is that cells here are sensitive to the orientation of a line in space. So that a neuron has a window on the world called a receptive field. And when that window on the world is excited with a line, a short line segment in which there is a light and a dark bar with an edge in it, or just even a light bar or a dark bar by itself, that is the optimal stimulus for a neuron. And why is this an interesting discovery? Because it was the first demonstration that underlying our behavior, underlying visual perception, there are neuronal bases. We see the world the way we do because there are objects in the world that are bounded by little line segments. And the reason we have object vision is because there are neurons in our primary visual cortex, not in the eye, but in the cortex, that are sensitive to little oriented line segments, which together bound or form the shape of objects in the world. So what this tells us is that visual cortex circuits create new properties from simpler inputs, such as circular center surround receptive fields of retinal ganglion cells get converted into orientation selective, line segment selective V1 neurons. How can this be? So many years ago, my lab did an experiment where we asked, can such properties be created in some other part of the brain? Where does the information come from that creates orientation selective cells? Is it an intrinsic property of the primary visual cortex, or is this a property that lives in the world? Because the world is made of objects. The world has line segments that bound the object. And is it that property of the world that can be utilized by the brain and by its mechanisms of wiring to create orientation selective neurons? So we did the following experiment, where normally in the visual pathway, information goes from the eye to visual structures in the brain to the primary visual cortex. Information from the ears goes from the ears to auditory structures of the brain and hence to the auditory cortex. We asked if we can make visual information go to the auditory structures of the brain, such as the auditory thalamus, and hence drive auditory cortex, would we get orientation selective neurons in the cortex that was made to hear, but is now being made to see? What would that cortex do with this input? We did these experiments in ferrets many years ago. And why ferrets? Because they have a visual system that is very remarkably similar. They're very visual animals. They're remarkably similar to humans. In fact, this is a painting more than 500 years ago made by Leonardo da Vinci. I actually went to the Czartoryski Museum in Krakow with my wife to see this painting. It is the dominant painting in this museum. And ferrets are, not only do they have great visual systems, but they also are born very early in development. So you can do manipulations to the ferret brain, such as remove the inputs from the auditory midbrain to the auditory thalamus, and which then allows visual inputs from the eyes to innervate the auditory thalamus. Ferrets are born at a stage in development roughly equivalent to the first trimester of gestation in humans. So we did these experiments. We took newborn ferrets. We changed the input pathways. Much later, we figured out why this experiment worked is because the gene expression patterns of neurons in the auditory thalamus are altered. Instead of expressing their normal complement of auditory pathway selective genes, they now begin to express visual pathway selective genes such as the ZIC genes, which allows retinal axons to recognize neurons in the auditory thalamus as potential partners. And thereby they connect to them, thereby allowing vision through the auditory thalamus to drive the auditory cortex. Then we let these animals grow up and we ask, what happens to the auditory cortex? And what happens is something remarkable. Like the visual cortex, in the normal animal, which has orientation selective cells and a map of these cells organized around singularities called pinwheels, which is a beautiful mathematical solution to the problem of representing all orientations in every patch of visual space within the cortex in a compact way. Similarly, the rewired auditory cortex in animals that are now have visual inputs driving the auditory cortex. These animals too have orientation selective cells that are organized as pinwheels. And the reason they have these cells and these kinds of maps is because the intrinsic connectivity of neurons within the auditory cortex has changed. Normally, within the normal auditory cortex, neurons connect to other neurons that have the same sound frequency representation. In the normal visual cortex, neurons connect to other neurons that have the same orientation representation across the cortex. In the rewired auditory cortex, neurons end up connecting in the same way as they do in the visual cortex, where neurons with the same orientation representation end up connecting to each other. And I remind you, this is a cortex that was never made to see. It was made to hear. But now, when it gets visual input, it alters its circuits and connections to process that input. Where is this information coming from? It can only come from the activity in these wires, which are in turn driven by the visual world. So it is the electrical activity in the projections from the auditory thalamus to the auditory cortex, now carrying visual information, that provides the drive for the neurons in the auditory cortex to make orientation selective properties and to connect themselves, change their connections in order to process that input, which leads to perhaps one of the most important questions to my mind in the field of developmental neuroscience. How does electrical activity shape cortical wiring? And this is a beautiful example of what people in machine learning or computer science called unsupervised learning. You don't need a teacher. The world acting through visual pathways helps wire the brain by altering the electrical activity in these pathways. And there are rules of wiring, there are rules of learning that can be used to understand how is it that electrical activity alters connections. One of these rules is called Hebb's rule. Hebb was a Canadian psychologist who lived 60 years ago. And he proposed that when one neuron repeatedly takes part in firing the other neuron, when you have temporal conjunction of pre and postsynaptic activity through what is called spike timing, then you increase the strength of connections between them. There are many molecules that implement Hebb's rules, such as NMDA receptors and then AMPA receptors. Complementary to Hebb's rule is another rule, which is that if you keep increasing the strength of synapses, after a while, all synapses will sit in saturation and networks will become unstable. So you need to scale down synapses, but keep the relative proportions of weights similar. That's called synaptic scaling and renormalization within local regions of dendrites, a phenomena that we have recently described a couple of years ago, that when some synapses go up in strength, within local dendrites, other synapses must come down. When a synapse increases in size, such as shown in green, and we can measure the increase in strength of specific synapses using very refined techniques, we can show that other synapses shown in red surrounding the increased larger synapses must go down in their size and strength. So synapse strengthening and weakening are locally coordinated. And together, these rules are implemented by many, 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 many molecules. The brain, our genome has 20,000 plus genes with many isoforms of these genes. And about 80% of these genes are expressed in the brain. And a very large number of these genes, the average synapse has more than 1,000 molecules. Hence, more than 1,000 genes are expressed in the teeny tiny space of a synapse that is 1 micron wide. And a large number of these genes and the molecules they make are dependent on electrical activity. And by imaging synapses in the living mouse brain, and by altering the expression of multiple genes, one at a time, or multiple genes, we can probe what are the molecules that influence synapse function, synapse structure, and hence synaptic plasticity. And we and others, through many experiments, have shown that a large number of molecules regulate synaptic plasticity. And together, these lead to the idea, and the first conclusion, very, very briefly described for you in this talk, is that brain circuits are wired during development by specific genes and molecules, and are shaped, brain circuits are shaped, brain synapses are shaped by rules and mechanisms of plasticity. And of course, our rewiring experiments and many other experiments in this field demonstrate that brain networks are crucially influenced by information from the world. But this is just the start of cognition. It tells us that cognition is deeply intertwined with the interpretation of the world that we find ourselves in. But our brains do not simply mirror the external world. The brain creates, it modifies the representation of the world in the service of cognition. So circuits wired during development are reconfigured to create dynamic representations that underlie cognition. And I'm going to illustrate this idea. If you remember anything from my talk, just remember this, that circuits wired during development are not static. They are continuously reconfigured through mechanisms of plasticity, such as I have described for you, in order to create dynamic representations. And I'll illustrate this with 3 examples briefly. Attention, how does the brain create and manifest attention? How does the brain create and manifest short-term or working memory? And even what might be the roots of reasoning? How does the brain reason? And how might we begin to understand that? Okay? Now, let's start with attention. Disrupting attention, disrupting cognition reveals the brain's attention networks. I started with the example of Anton Rederscheidt, how a stroke in the right posterior parietal cortex leads Rederscheidt to not attend to the opposite world. It's a very well-described phenomenon. It's called left spatial neglect. And here are other patients who had similar strokes as Rederscheidt. You give them a model, draw a clock face. They draw only the right side of the clock. They ignore the left side. It's not that they can't see, it's not that they can't draw. They simply deny that that left side exists. You give them such pictures, they draw the right side. They ignore the left. They ignore the left side. So it's a deficit of attention. And it says that this part of the brain is really a very crucial node for attention. Hence, studying this part of the brain may give us clues to understanding this deep substrate of cognition. Attention filters and focuses information from the world, is crucial to cognition, as you can see at a glance from these drawings. How are such internal states? It's a deeply private condition, attention. When you are looking at your screen, I have no access to what you are paying attention to. Maybe you are dreaming about something else. So how might we even study? How might I study a private internal state that only you have access to? This is the deepest challenge of studying cognition and understanding its neural basis, but we can. We don't often have direct access to the human brain. We don't want to do invasive experiments. We can do some experiments. We can image the brain. We can record electroencephalograms. We can do non-invasive experiments. But to really understand brain mechanisms, we have to go into the brain. And many animal models have been used. An animal model that my lab and many other labs uses, I already mentioned ferrets to you. We work with monkeys as well. But the animal model that allows us the most access to the brain is the mouse. And mice, of course, diverged from humans in a line that is maybe 100 million years, 50 million years ago. Nonetheless, they have, I don't know how many million years ago, many million years ago. So the mouse brain has features that are common to the human brain. A mouse brain is 1,000th the size of a human brain. A mouse brain has 30 to 50 million neurons, as opposed to 80 billion neurons that humans have. But the mouse brain is by no means a human brain in miniature. But the mouse brain has sensory areas such as the visual cortex, shown in blue, or the auditory cortex, shown in yellow, or the somatosensory cortex, shown in red, that Neeraj Jain studies. It even has early variants of the higher areas, such as the parietal cortex that I've been talking about, but in much smaller form, and the frontal cortex that I will talk about, but in much smaller form. So at least the core functions of these higher areas can be studied with appropriate caveats in mice. And mice, of course, allow the full power of modern molecular genetics and imaging technologies to be brought to bear on neuroscience or studying how the brain works. We can create mouse that express certain fluorescent reporters whose activity can be imaged using lasers and multiphoton technologies in order to understand how hundreds, thousands of neurons, how specific cell types process information and interconnect with each other to give rise to the circuits of cognition. How might we study attention? Attention is something that pervades cognition. You are stopped at a red light, particularly if it is a little bit cloudy or a little bit foggy. You take in information from your eyes. You need to decide, is it red, is it green? You make a decision, and if it is green, you go. And so all these aspects of cognition, perception, decisions, action, or choice are enabled by attention. If you are not attending, you will not be able to make any of these judgments, any of these cognitive judgments. This is an arbitrary rule that human beings have made, but they are very good at it, that red means stop, green means go. We can take mice and we can also train them on arbitrary rules. We can train them to attend or not, and we can ask, how did the brain mediate this rule and the attention that went with it? So here's the idea that we can train mice to respond to one set of stimuli, in this case, horizontal gratings, and ignore another set of stimuli, in this case, vertical gratings. When the mouse is given a cue, and we are like circus trainers. By we, I mean my students and postdoctoral fellows. We can train mice to do amazing things, or at least simple tasks in very effective ways. You cue the mouse. Now there is a stimulus coming on. Then you give it one of these two stimuli for a couple of seconds, and that's important. Hold that in mind, that the time space of these experiments is a couple of seconds of stimuli. And then you bring a lick spout forward, and the mouse should lick when it sees the target, and it should not lick when it's not the appropriate target, so that when it sees horizontal grating moving up, that's a hit, and you give the mouse a reward. And this is the essence of reinforcement learning, that by a phenomena called reinforcement learning, where you reinforce the appropriate response, and you do not reinforce or you even punish an inappropriate response, such as responding to an incorrect or non-target stimulus. The mouse should not lick. If it licks, you de-reinforce it, and the mouse eventually learns with a high level of confidence over a few weeks of training that this is a go and this is a no-go stimulus. I'll show you a very brief video. So here is the mouse. I hope you can see this. Here is the lick spout. Here is the screen at the back. The mouse will see a visual stimulus, a target, and then a no target, and you'll be able to see that the mouse responds or not in the right way. This is a highly trained mouse. And so here we go. So here is the mouse twitching, and then here is a stimulus, and then the lick spout comes forward and the mouse licks. And so that was the target stimulus. Here is a non-target stimulus, vertical grating, and the mouse does not lick. So in this way, we know that the mouse has been trained by reinforcement learning to do this task. And now how do we control its attention? This is what we do, that we give the cue, we give the stimulus, and then we give a reward in order to engage the mouse. But in certain blocks of trials, we do not bring the lick spout forward. We do not bring it forward, so the mouse learns very quickly. I'm in a situation where I am not going to be rewarded. I have no interest in doing this task because it is a useless expenditure of energy. There is nothing to lick, and I will not be rewarded. So the mouse switches off, and that's called the passive condition. Is the mouse really engaged, attending or not? You bet, because we can monitor the eye's pupils. And when the mouse is attending, the pupils dilate. And so in the engaged condition, the mouse is able to lick and does lick, and therefore we can distinguish between the hits and the false alarms, which is called this metric called D prime. It is a well-known aspect of learning theory as to how to measure learning. When the mouse is in the passive mode, it cannot lick and there is no response and the mouse is disengaged. What happens to the brain? We can use multiphoton, 2-photon imaging. This is high resolution imaging technologies that my lab and other labs have developed to look at the activity of neurons, hundreds of neurons simultaneously in the primary visual cortex and the posterior parietal cortex that are expressing this genetically encoded calcium indicator. This is why we use mice, is because we can make mice that are expressing the genes that make this molecule that can be used as a source of light. Because a laser, these molecules absorb light, and the more calcium that enters the neuron due to the electrical activity of neurons, the more light that can be emitted and imaged. So here are about 70 neurons in the space of primary visual cortex in a mouse that is performing this task. And each flash of these neurons is a spike. And we can image these flashes at very high resolution in the primary visual cortex and the parietal cortex. And when we analyze the responses, what do we see? This is what we see, that neurons in the primary visual cortex respond to whatever stimulus was presented. But neurons in the parietal cortex, in this higher area, respond when the animal is attending, regardless of whether the stimulus is present or not. The same stimulus can lead to a response or not, depending on whether the animal is attending, on the internal state of the animal. So here is a V1 neuron that likes horizontally oriented bars. And I started by telling you visual cortex neurons respond to different orientations of stimuli. Here's a neuron that responds to horizontal stimuli. Other neurons respond to vertical stimuli. When this neuron responds to horizontal stimuli, it responds irrespective of whether the animal is going to be rewarded or not. It is an automatic response whenever the stimulus exists in the world. Here is a neuron in the posterior parietal cortex. It responds to the horizontal stimuli, but only when the animal is engaged or is going to be rewarded. When the animal is not going to be rewarded, the same stimulus has no response. So parietal cortex neurons are strongly attention dependent. I remind you that the visual stimulus is identical during engaged and passive trials. Only the internal state of attention is different. It's only the mouse's mind that says, I should switch off because these guys are not going to give me any reward or any water. And these neurons stop responding. We can go further. We can switch the contingency. We can train the animal to say that the horizontal stimuli means you will be rewarded, to now that the vertical stimuli means you will be rewarded. We reverse the task contingency, as it's called. It's as if we were trained that go means stop, that red means stop, green means go, and now we flip it. Now, red means go, green means stop. For those of us who have been conditioned on these colors and on these symbols, artificial symbols, it is deeply disturbing, and it tells you the power of supervised and reinforcement learning in our lives. But for mice can actually learn this reversal very quickly. And when they do, you can show that their response reverses. Now they're responding to the vertical stimuli with licks. What happens to the brain? In the primary visual cortex, nothing happens. A neuron that was responding to horizontal bars continues to respond to the horizontal bars even when the meaning of the bar has shifted. In the parietal cortex, though, there is a remarkable switch that's dependent on the meaning of what these bars is. A neuron that used to respond to horizontal stimuli now because the vertical stimuli give it reward, is the same neuron. And that's the other reason we use mice. We can image that same neuron over weeks at a time in the same animal, in the same part of the brain, because we can train the animal, we can retrain the animal, and we can keep imaging every day that same neuron. So it's a remarkable way to understand at the level of a single neuron, which is 1/10 the width of a human hair, and its synapses, which are 1/100 the width of a human hair, what is changing as the animal is learning and relearning. These neurons in the parietal cortex encode the meaning of the stimulus. They encode horizontal stimuli when that gives reward, and they switch to encode vertical stimuli when that is what is associated with the reward. So they switch selectivity. And indeed, this switch is very remarkable. In the visual cortex, the original or the reverse condition, there is no change in the numbers or the properties of neurons. In the parietal cortex, in the original training condition, many more neurons responded to horizontal. Now, many more neurons respond to vertical. Why? What does this mean? It means that the parietal cortex transforms vision to action. From the stimulus that is seen by primary visual cortex, the meaning of that stimulus requiring attention is transformed and fed to the motor cortex in order to execute the action. So V1 neurons reflect the sensory stimulus. Parietal cortex neurons respond to the rewarded stimulus and convey a decision. Because before you can act, you must decide this is worth acting. You choose a motor plan, and attention strongly modulates parietal responses, and it leads to what we think is one of the deepest ideas about attention. Why attend? What is attention? Attention reflects action selection. You attend when the meaning of a stimulus has to be converted to an action that, at least in the context of tasks like this, are rewarding. That is what attention is. Attention is action selection. So task switching involves top-down gating and illustrates synaptic plasticity that reconfigures V1 to posterior parietal circuits. In the original condition, there are horizontally selective V1 neurons that drive parietal cortex neurons, which drives motor cortex. Now, when we switch, due to the relearning, which involves modulators that I'm not going to talk about, now the vertical stimuli end up driving the parietal cortex neurons, and this is a flexible reconfiguration of the V1 to parietal cortex circuit. And it illustrates the importance of plasticity and reconfigurable circuits for phenomena such as attention and relearning. There is a dynamic coding of choice. I'm running short of time, so I'm going to very quickly go through this. We can build decoders. When a part of the brain has hundreds of neurons firing, how do you make sense of it? You use statistical tools, and we infer. These are the same ways, mathematical ways, by which another brain region that is looking at this brain region, that is getting input from this region, such as the motor cortex getting input from parietal cortex. How does the motor cortex infer? What is the parietal cortex telling it? One way to think about that comes from statistics and the new statistics of machine learning, and it's called decoding, where you can decode, you can build a statistical separator for 100 neurons and ask, for these 50 neurons, are they separable from the other 50 in terms of what is being encoded? And if you build an encoder for a certain point in time for 100 parietal neurons and you ask, does the activity of these 100 neurons persist in a way that signals a stimulus throughout time? Well, if you build a decoder for each point in time, of course it does. But if you build a decoder for one point in time and you ask, how long does that set of neurons continue to represent that stimulus and hence encode the action? The answer is only for a couple of seconds. And I remind you, this is the space of the task over which the stimulus was presented. So posterior parietal neurons dynamically encode choice. And that's what this means. When you decode hit from correct rejects, for example. In contrast, visual cortex neurons have very static encoding of choice. A visual cortex neuron responds to a horizontal stimulus or a vertical stimulus, and it does so for the entire duration of the task and action. Hence, we call this static encoding. So posterior parietal circuits reflect decision and choice dynamics of the task in the context of the timing of the task. And it tells us something very profound, that plasticity and computation are encoded in the same parietal cortex neuron. This is one of the deepest ideas that underlies cognition. Unlike your computer, where you have a processor and you have a memory, and a lot of time and effort and energy is spent in shuffling data in and out from your memory, your hard drive, your random access memory, into the CPU and back. In the brain, a parietal cortex neuron is a computing element. It converts vision to action. And it is also a computing element that is dynamically changing because of plasticity. And you can see at a glance the time course of that dynamics. So it is as if the CPU in your computer is dynamically changing based on what has just happened or what it is required to do. The hardware is continuously being changed by the software, if you wish. And this is one of the deepest differences between the biological brain and a silicon computer, at least so far. Very quickly, short-term or working memory is another crucial state that underlies cognition. How might we study this? We can take the same task and we can add a delay between the stimulus and the action. And adding this delay simply delays the animal's action. We can add delays of 3 seconds or 6 seconds. The animal's hit rate doesn't change too much, but the animal makes more mistakes. The false alarm rate, meaning that it responds when it shouldn't, goes up, but still the D prime, or the difference between these in units of standard deviation, is still very high. Here's a video. Here's again a mouse. Here's the lick spout. You will see a stimulus, and then the stimulus will go away, corresponding to the delay, and then the lick spout will come forward and the animal has to lick. Okay? So here is the horizontal stimulus during the stimulus period. Now we are in the delay period, and now the lick spout will come forward and the animal licks. Okay, so the animal can be trained on this task. And our goal is to ask what happens in the visual and the parietal cortex, but also in the motor cortex, as the animal has to incorporate this working memory into its behavior. And I remind you, a great amount of cognition is working memory. If there is anything you are retaining from my talk, is because your working memory over the last several tens of minutes has been held in your brain. If you did not have that, anything I'm saying will make no sense to you. So we are recording the activity through wide field imaging of neurons in these 3 areas now: a visual area, a transforming area, and an action area, the motor cortex. And what do we see? We see something very remarkable, that it is the activity in the motor cortex, shown in red, that shows the delay period activity. And you see that at a glance here for delays of 6 seconds. The stimulus comes on, shown here. There is a delay period. I remind you, during that delay period, there is no stimulus in front of the animal. There is nothing in front of the animal. There is only the animal's memory, which we cannot see, which we have no access to. But now, if we record the neurons, we know that the animal is holding in memory the action that it is supposed to perform. And that is why the motor cortex has the delay period activity. And so this is an internal state. It is an internal representation of time that the motor cortex holds. We can describe this, and then when the lick spout comes forward, the animal licks. we can do very quick analysis of dynamics to explain this. If you have one neuron, you can show this activity goes up and down in time. If you have two, if you have three neurons, you have to plot the activity in three-dimensional space. If you have 100 neurons, you have 100 dimensions. It's impossible to represent this pictorially at least. So we do dimension reduction. We extract the components of these hundreds of neurons into two, which are two factors, and this is called a reduced dimensional space. And we can plot these two dimensions against each other to see how is the task, the stimulus, the delay, and the action represented in the space of 100 neurons, in this case, 400 neurons in visual cortex. And here's what we see, that there are two kinds of components. One kind of component in primary visual cortex that represents the horizontal stimulus, another component that represents the vertical stimulus. There is no signature of the delay. When the stimulus comes on, one neuron or the other fires, and then they go back. But this is not what happens in the motor cortex. In the motor cortex, you see beautiful signatures of the encoding of each of these task states. And you see the signature of cognition in the trajectories of these population vectors. And here are the two principal components. And here is one component of motor cortex neurons, where the neuron begins to respond when the stimulus comes on, shown in blue. Then in the delay period, it is holding that activity in what is called a basin, an attractor basin. And then a second set of neurons, or a second component of neurons, then executes the action. And this has been well described as arising from specific networks called recurrent networks with a role for inhibition in keeping the activity within limits. So motor cortex neurons, shown in red, build their activity during the stimulus period itself, demonstrating rapid action planning. And then they hold that activity, and then another set of neurons executes it. And you can see this here. One set of neurons comes up during the stimulus and holds that activity as the internal representation of working memory and of time. I must lick as soon as I can. And then another set of neurons executes that lick exactly as you saw in the dynamic trajectories of the principal components. So motor cortex neurons predict a central role for motor cortex as a crucial component of cognition in all components of task performance, in the action planning, in the memory, and in the execution. What does this mean for networks and for plasticity? The brain has many regions that go from vision to intermediate steps to action. But learning and executing a visual discrimination task, like we have just taught our mouse, involves rapid routing of information, even as the stimulus has come on. Oops. Even as the stimulus has come on, it's just a visual stimulus. The mouse has already decided, this is the stimulus to which I must lick. So the motor cortex activity shown in red has already gone up. This is the rapid transfer of information from the visual cortex to the motor cortex, and is being held in the motor cortex as working memory. Memory is held where it has to be used. And that's what this says. So there is a rapid transfer of information, not meandering through all these areas to the motor cortex, and is held there till it is used. So flexible dynamic reconfiguration of pathways and networks underlies fundamental processes of cognition, such as attention, decisions, learning. And we submit they likely lead to some of the highest attributes of the human mind. And at this time, I need to ask my host, Neeraj, how much time do I have? Do I have 5 minutes?
Yes, absolutely. Please go
last
ahead.
Component of my talk on reasoning, and I'll take 5 minutes.
Yes, sure. Go ahead, please.
All right. So what does this have to do with our deepest roots of cognition? So there was a philosopher of science, there is a philosopher of science who used to be at MIT, and in 1972, he wrote a book called What Computers Can't Do. His name is Dreyfus. And Dreyfus critiqued machine intelligence. Dreyfus said that old-fashioned artificial intelligence based on symbolic representations to produce general intelligence is bound to fail. And 20 years later, in 1992, Dreyfus issued that same book, but with a new introduction. It's called What Computers Still Can't Do. And so I'm fond of saying that's one way for this is not a good example for our younger colleagues, how to get 2 publications out of 1. But in the last several years, we have seen a remarkable progress in understanding artificial neural networks. And what they are telling us is that specific models speaking to specific components of behavior and even cognition may be successful, and they incorporate connections. So, and they, to me, remarkably, some of the most interesting work speaks to the roots of cognition, speaks to the roots of reasoning. And in the next few minutes, and I promise I will end in a few minutes, what does this mean? How can we think of reasoning? Dreyfus talks about artificial reason being not incorporated in machines. Does the mouse brain have signatures of reasoning? Can we use this simple task to ask, if not reasoning with a capital R, at least reasoning with a small r, a logical sequence of actions that optimizes behavior? And we think the mouse's brain does show instances of reasoning. When we look at error trials, when the mouse makes a mistake, why does it make a mistake? This is a highly trained animal. It depends for half of its water intake on the task, yet it makes a mistake sometimes. And the mistakes are of 2 kinds. The mouse sometimes licks when it shouldn't. So when there is the non-target stimulus, most of the time the mouse does not lick, it correctly rejects. But occasionally it gives a false alarm, and that's called positive error. And a mouse sometimes makes a different kind of mistake. When it should lick, it doesn't, and that's called a miss. So why does a mouse lick when it shouldn't? And one possibility is that maybe something went wrong with its visual cortex, and so it saw things incorrectly, and that may be true. But a more interesting possibility, but if you look at the visual cortex responses during false alarms called positive error, they don't change too much compared to when there is a real stimulus for a hit. It is the parietal cortex responses in green and the motor cortex responses in red that are really high. And so the mouse's brain is generating the signals to lick when it shouldn't normally. And this leads to one of the deepest ideas of reinforcement learning. I remind you, I told you, this mouse has been trained through reinforcement learning, through classic reinforcement learning. And the deep idea of reinforcement learning is that error trials may arise from exploration at the cost of exploitation. Suppose you are driving from your home to your university, and you have chosen a path, and you take that path every day, and you have been told this is a good path to take. How do you know this is an optimal path? You will never know unless you try other paths. So you need to have a policy. I'm going to try not this path, but that path, or go there. Even though Google Maps tells me this path is the best, unless you try something else, you will never know. And that's called exploration. And you need to have a value function. You need to evaluate the cost or the benefit of having that policy of choosing a different path, and that will be your time. I took less time today, so now I'm going to take this other path. So this is how exploration enables optimization, but is sometimes at the cost of exploitation, which is continuing to do something well. The ideas of reinforcement learning, when applied to machine learning, I believe, in the last few years have led to some of the deepest insights into the roots of reasoning. And I'll tell you in 2 slides why I think so. So machine learning has led to remarkable successes. Most of machine learning is about classification problems. You know, if I spell Neeraj wrongly in my Google search, if I say Neiraj, well, Google has a set of classifiers similar to the decoding classifiers I told you that will say, did you really mean Neeraj? Fine. But the most interesting aspects of machine learning come about when computers or artificial neural networks have been trained to do interesting tasks. And some of the most interesting tasks within the space of computation are games. So artificial neural networks have had remarkable success in games with perfect information and deep search. What are these games? These are games like chess. These are games like Go, where there are 2 players, where there is zero sum. If one wins, the other has to lose. They're deterministic, at least in principle. Every move can be figured out. They are discrete. I move, then you move. They are Markov, meaning that what you see in front of me perfectly defines the state of the game. When a new player walks onto a chessboard that is halfway played, that player doesn't have to know how you got there. Based on what he or she sees in front of her, she can continue to play the game. That's called the Markov process, as opposed to non-Markov process, where a lot of history has to be considered in giving a reward. So this leads to perfect simulation of game states. It's been 23 years since a machine learning artificial neural network beat the reigning chess champion of that time, Garry Kasparov. So that's old news. Computers already have superhuman performance in games like chess, but this was achieved by very specific hand-tooled learning rules. In 2016, I believe there was a transition. There was a singularity in our understanding of reinforcement learning, where a group in London called DeepMind built a program called AlphaGo that beat the reigning Go champion. For those of you who have never played Go, it is a board game with 19 by 19 squares, etc. And the game of Go has 10 to the 170 moves. Chess has 10 to the 120 moves. These are enormously complex games. Just as a matter of comparison, the atoms in the known universe are 10 raised to the 80. And then in 2018, in a version of DeepMind that is a general purpose game playing machine, built entirely by learning to play itself, their program AlphaZero beat another superhuman program called Stockfish, because computers were beating humans a long time ago in chess. But even the best program of that time from 1.5 years ago, AlphaZero, based on reinforcement learning, beat Stockfish. What does DeepMind's programs do? Very quickly, DeepMind's programs use value networks to evaluate bird positions and policy networks to select moves. I gave you a little flavor of policy and value in my example of driving to work. And their deep networks are trained by a combination of supervised learning and from humans and reinforcement learning from games of self-play. The computer plays against itself. And in addition, they combine Monte Carlo simulations. There are 10 raised to 100 possible options as the game progresses. You can't search them all, but a principled way of searching through that space combined with the policy and value network leads to remarkable performance. In a second paper from 3 years ago, they removed the human intervention at all and just made the computer play against itself with just the rules of how should each chess piece move and what does it take to win that your king remains standing. By playing hundreds of thousands of games with itself, the computer beat its own master program from the previous year. And then 1.5 or 2 years ago, they built a general reinforcement learning algorithm that masters many games, including chess. Since most people in India know how to play chess, I'll give you a brief flavor and then I'll conclude. So this is a computer that has just been told Here are the pawns, the rook, the bishop, this is how they move. And when you win, when the other king is under checkmate. After 300,000 steps, this program AlphaZero beat in teeny tiny improvement Stockfish, which was the winner of the chess championship from 2016. And in the game of Go, similarly, AlphaZero, which is this program, general reinforcement learning program, beat the previous program. And AlphaZero beat Stockfish not just 1 or 2 times, 30% of the time. How does AlphaZero play, you might ask? Just for the first 2 moves, millions of people the world over have certain moves that they use, and AlphaZero, of course, has those same moves. But within a move or 2, AlphaZero, within 4 to 5 hours of simulation, playing against itself. I remind you, these are not simulations on your laptop computer. These are on tensor processing units and graphic processing units, and tens of them that these games are being simulated, okay? But within 2 moves, this program beats the previous master program 25, 30% of the time, and the others are draws. Here's a quick example from an accompanying piece to that paper in Science. Here is a chessboard. The red moves are rejected by AlphaZero outright. They are moves that I, as a novice chess player, might well choose. It homes in on these 2 moves, and it finally picks the blue move, which to most eyes would seem the most risky move, because this spot has 2 forces on it from black and only 1 from white. But it still picks that move with an eye to the future because it opens up lanes. In fact, Vishwanathan Anand wrote a blog piece on this paper. And Anand says that it's amazing that it figured out everything from scratch, and that is scary and promising. I would like to think that it should be a little bit harder. He himself has spent decades trying to figure out moves, and it figured it out within 4 hours from the first move, what will be the path to victory. A more perceptive commentary comes from Garry Kasparov. And Kasparov, of course, was the world chess champion. And he wrote a piece called Chess, a Drosophila of Reasoning, that accompanied the DeepMind article in Science from 2 years ago. And this is what Kasparov writes. I admit that I was pleased to see that AlphaZero had a dynamic open style like my own. The conventional wisdom was that machines would approach perfection with endless dry maneuvering, leading to drawn games. But in my observation, AlphaZero prioritizes piece activity over material, meaning positioning over how many pawns do you have of the opponent, preferring positions that to my eye look risky and aggressive. Programs reflect priority and prejudices of programmers, but because AlphaZero programs itself, I would say that the style reflects the truth, reflects the truth. What does Kasparov mean? And this is my last point. Where does reasoning come from? People have argued reasoning is invented by the human mind. And others have argued reasoning exists, that the rules of logic exist and we simply discover them. If we have a computer, if we have a machine learning program, an algorithm, that simply from the rules of how chess pieces move, it figures out the logic of the game through reasoning, it must mean that this is the logic of the universe. This logic exists outside of us, and we figure it out, we discover it. And this, to my mind, says in perhaps a reasonably profound way, though it needs more analysis, that our cognition reflects the properties of the universe we find ourselves in. So in conclusion, the brain wires itself, I started out by telling you, it creates its own architecture using mechanisms of plasticity and specificity, and it takes information from the world in order to wire itself. in addition to the genes and molecules that it intrinsically expresses, but they too are shaped by electrical activity. And then I told you this architecture is flexible and reconfigurable. It enables cognitive states such as attention and functions such as action selection and decision making. And this is what happens in areas such as the parietal cortex, which are core nodes for attention as a window into action selection. And then I told you the neural dynamics in brain regions through the trajectories of populations of neurons navigating through space and time, create internal representations of tasks, such as working memory and choice-based actions. And this I illustrated with data from the frontal motor cortex. And finally, I took off and argued for you that plasticity, reconfigurable architectures, implementing the laws of reinforcement learning can really incorporate the core ideas of reasoning and potentially higher mental functions. I thank you. I apologize for going over, and I am going to end by acknowledging the people in my lab at this time, other people who have contributed to these experiments, who have moved on, most of them to their own labs, and various collaborations. I apologize that we have many collaborations, but I could only fit a few whose work directly speaks to the topic of my talk today. Thank you very much. And we have several, okay.
Well, thank you, Professor Soor, for that deeply engaging talk and taking all of us all the way through from very, very basics of neuroscience to the complexity of brain function, which we all call cognition, and how it fits into the modern world of machine learning and where it is going. It is amazing to hear you, and it didn't at all feel that you have gone over. I thought you could have been speaking for another 1 hour and we would have been so much enraptured. And the people who joined us little late, I started out by saying that Professor Soor is among the top neuroscientists in the country, and anybody who has done any neuroscience would know him. He is the Newton Professor of Neuroscience and the Director of Simon's Center for Social Brain at MIT in Department of Brain and Cognitive Sciences, which he headed for many, many years and made the department what it is today. I myself would just pass on few questions from the audience before I invite other panelists to give their comments. And some of the early questions, and if this audience is still there, Subra had asked, and this is the toughest question in my opinion, perhaps not for you, is why does the left hemisphere of the brain represent the right side of the face? Is there some benefit to cognition? Would you like to comment on that?
Yeah, I think many people have speculated on this, and I believe that our brains are products of evolution, and the short answer is that both for sensory systems as well as for motor action, there is some benefit to having crossed representation. The lens in the eye, which is a remarkable feat of biological engineering, that you have proteins that are transparent, so transparent. So you can only make so much of them, and you can't make two lenses. You make one lens, and the lens inverts rays when it comes into the eye. So there's a very important physical property of light that has to be utilized, or that is reflected in the brain. And so vision evolved very early. It evolved independently through multiple times, but all vision in every species involves lens that inverts rays of light. And that directly leads to the idea that the right side of space is represented in the left side of our body. Secondly, when you move in response to that a sensory stimulus, then oftentimes you need to coordinate activity in the two sides. And it makes sense for each side of the brain to influence the opposite side alongside modulating the same side. These are not very satisfactory answers, as in a lot of biology. An appeal to evolution is a just so story, but it seems plausible that such evolutionary adaptations have held on into very, very high level phenomena, such as the way that the human brain works.
Well, thank you. That's a very, very interesting angle to this question that, of course, is foremost in everybody's mind. And as you said, we are essentially scrambling for explanation to a perfectly evolved system, which has evolved over millions of years. I was just wondering, since you said the primary reason for this is the way the lens, so that essentially means vision. But what about the other systems? Because one would think that tactile system is probably more primitive than vision in certain sense. We still have a crossed pathway for that, or motor system, which partly, at least, if not majorly, in fact, majorly controls the opposite side.
I won't speculate any more than I have to, but
okay.
It is possible that a lot of brain processing is geared towards the final action or the motor response to inputs. And in fact, there is an influential set of ideas that says that the real task of the brain or the mind is to act on the world, not just to take in sensory input, but sensory inputs only make sense in the context of acting on the world. In fact, my own view of attention or of internal states is not too far from that view. Why attend? And the only reason, one reason to have internal states is to facilitate acting on the world. And that may be facilitated by having crossed pathways. So then everything else falls along.
Well, thank you. I'll pick one more question before I pass on to my colleagues here. And this is because you do have tremendous interest in autism and you have published remarkable papers in that. So the question here is from Kunj Bihari. How does cognition get affected in autism spectrum disorders? Are pathways affected?
Yes. So I should have said that why should we study the neural architecture of cognition? There are at least three reasons. First is, of course, to understand ourselves. And that's one of the highest quests of science. Why are we the way we are? Why do we think or act or you know, how do our brains work the way it does? But second is to understand, have a deeper understanding of brain disorders, not just at a very reduced level of genes and molecules, but at the levels of functions and circuits, perhaps in ways that span many different immediate etiologies, such as this gene or that gene, which we also study. But can we have a broader picture of why cognition and disorders of cognition happen, which is a great many neuropsychiatric disorders are disorders of cognition. And of course, a third reason to understand cognition is to build better machines, the next generation of machines that whether you favor such machines or not, it has been a quest of humankind to build better machines. So autism is a very genetic disorder. And there are genetic mutations that have been linked to autism, but a lot of autism is about complex genetics. It's not just a single gene. And the more complex the genetics, the less likely that we will have a very reductionist view of how can we understand it, how can we treat it. And so we need ideas such as this at the level of networks, at the level of brain regions, at the level of plasticity in reconfigurable architectures and circuits in order to understand how brain regions give rise to the cognitive components of autism. So that's just a broad answer to how does this work fit with autism and what do I see as the future of the field.
Thank you, thank you. I'll pass on to Dr. Vidita Vaidya here for further questions. Vidita?
I'm right here. What a terrific talk, Mrigank. I mean, it was absolutely thought-provoking, especially the ending, which opens up a whole series of questions about what is reality and what is eventually our ability to come up with rational thought process. Do we just acquire it and it's something that exists out there in the world? I had a question that continues off from where Neeraj ended up, which was, is synaptic scaling and renormalization that you talked about something that is commonly disrupted whenever you have dendritic structure abnormalities, either in mouse mutation situations or when there's been, change in a neuromodulatory pathway? Is this a common target via which you actually end up with neurodevelopmental and neuropsychiatric like disorders? I wanted your comment on that.
Yeah. So we can speculate on it. People haven't studied it directly at the level of dendritic processing, but I've written a couple of articles on this as to how a failure of scaling or a renormalization is a potential reason for how the genes of autism or the genes of neurodevelopmental disorders might be active. So when we have autism, even if it is due to a single gene, such as Fragile X disorder or Rett syndrome, which I study as well, or Angelman syndrome and so forth, overall, the brain is built fine. It's not that people have remarkable difficulties in this or that or the other. But there are specific difficulties. There are specific cognitive dysfunctions. There are specific difficulties in learning. And a unifying view is potentially that plasticity involves Hebbian mechanisms combined with synaptic scaling and renormalization, and a deficit in renormalization. And the molecules of renormalization, such as ARC, are at the nexus of many different genes of autism. So as a putative unifying view of how mechanisms of plasticity may be affected in autism, it is a reasonable hypothesis.
I'm going to take one of the questions from the audience and put that out to you. It's coming from Shweta, where she says, is the set of neurons activated during the attention task depend on the nature of stimulus? And I'm going to tag on a little bit of my own question to that to ask whether punishment in reward, when you flip between punishment and reward, does the PPC neuron activation in terms of classes of cells or even in terms of the manner of response change when it's aversive versus appetitive?
So our experiments and of several other labs shows that it is the same neuron that is now flipping its drive, because we can track that one neuron, and we have done this for tens of neurons through multiphoton imaging and imaging the same neurons over time as on the 2-photon microscope, the animal is being trained or retrained. So at least for certain neurons, it is their inputs that are being rewired. No question about that. That is the only way that this neuron can change. So how is that so? And one possibility is that it is the V1 to parietal inputs. By V1, I'm using this broadly. We have some ideas, we have measured the V1 input, but there are other visual inputs to these parietal neurons. And clearly the strength of those inputs is being modulated, and is likely being modulated by the reward signal. And so that is why in my slide I had neuromodulators, which are the stand-in for reward signals, such as dopamine, but importantly, norepinephrine and acetylcholine, because as you go back in the brain, as you know, dopamine is much more important for striatal and frontal functions, but the other neuromodulators come into play and much more prominently, at least relatively prominently, for more posterior cortical areas. So the role of these neuromodulators, and we and others are looking at the effect of norepinephrine as signaling reward and punishment as well. So certainly there are punishment signals, there are reward signals. The switching of those signals provides a powerful driver to modulate the strength of sensory inputs onto individual parietal neurons. Whether there are other parietal neurons that are also being brought into play, chances are they are, but we have less information on that.
I'll go to a question that links up to that in a sense. And the question comes from Akshay, who says, in mouse task switching experiments, have you looked at the age of the mouse and how does that affect the behavior?
Sure. So certainly older mice don't learn as well as do younger mice. So we always use adolescent or young mice and so forth. And the more wild type, the better. The more inbred they are, the more complicated. And of course, mouse strains are all inbred strains, but still, and so forth. So there's no question there are many, 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 many drivers of learning and the propensity to learn. And age is one of them for a variety of reasons, including the effectiveness of synapses and the effectiveness of neuromodulatory signals to encode learning.
So I'll end before I hand over to Arun with a question that I wanted to ask, which ties into the latter part of your talk with machine learning. This case, it was with a game, but when we see more and more machine learning coming into reality and use, what about implicit biases that get coded in based on the manner in which these programs are originally set up, or in terms of the information that is fed to them for which eventually they use for reinforcement learning? What does that do into our world in terms of potentially amplifying existent biases that exist world over? And what are your thoughts on that?
Oh, it's a very, very deep and interesting question, an important question. There has been a lot written about it. Any artificial neural network is, when it's trained up, is only as good as the training set. And so even Kasparov, who's actually a fairly reflective man, says that oftentimes neural networks reflect the biases and prejudices of people who make them. And that should be defined broadly, including the training sets that you use to train networks. It has been famously said how face recognition software misclassifies Black people, and because the training set doesn't have enough Black people. And this is a terrible travesty, of course. So there is no question that machine learning needs to be integrated with social justice. Coming back to Kasparov, the interesting thing in this example that I gave, which is why I gave it, is that the only examples that this algorithm has are the examples it has generated. So it keeps playing with itself, playing against itself, and generate the example. And of course, chess or Go or board games are a very rarefied, very, very narrow example of real world application. In the real world, there have to be representative examples that represent all of humankind or their condition, of course.
Thanks, Ragang.
Thank you. Thank you, Vidita. Thanks a lot. I'll just read out certain comments which I can see in the YouTube chat from the audience. Amazing talk. Thank you very much. Great talk. Captivating lecture. Very exciting, very enlightening talk. I mean, this just goes on and on and on. This is something that you are not able to see as you're going on. I'd like to invite now Arun Shripati from CNS, Indian Institute of Science, Bangalore, for his comments and taking the questions further. Arun, please go ahead.
Yeah. Reconfigures, thank you for a really fascinating talk. Even though I've been in the field for a while, I also find it very inspiring and interesting. So I thought I'll, and also I want to add that there's a lot of interesting questions which also I think reflect the kind of talk that you gave, because there's a lot of deep questions coming from the audience as well. So maybe I want to start with one of the audience questions and maybe then ask one of my own. Actually, another person also with your same name, Mrugank, has asked what is the advantage of dynamicity that is observed in the PPC, and wouldn't this dynamicity result in more errors in terms of replicability?
Mm-hmm. So it's not a question, what is the advantage of dynamics? It is the way that neurons, via their properties, encode a dynamic world. The world is changing. The world of the task is changing. There is a stimulus, there is a delay. Now you have to respond, then you get a reward. So there are epochs in the task. And the animal, or we drive, we get into our car, or we walk from point A to point B. We achieve a goal, we get some reward or not, or whatever. So all of life, the world itself, plays out in time. Hence, the representation of that world has to represent that time. And this is what neurons do. And a single neuron can't do it. There is too much complexity in the world. So you need groups of neurons. These groups of neurons must follow rules. They must follow rules of mathematical representation, of statistics, of logic. That is the only way that there can be a systematic representation of the world in space and time. And that is how one part of the brain can be interpreted by another, or we as experimentalists can interpret the activity of one or more parts of the brain in relation to the world or to the task. So that's the logic of using the computational ideas of trajectories, of population coding, of dimension reduction as simplified statistical methods for understanding populations of neurons and the dynamics of neurons are necessary components of understanding the brain.
So I wanted to add to that by asking a question that has sort of always struck me when I see recordings of decision signals from different areas in the brain. And I always wonder, do you think it's going to be possible to find the origin of these signals, for example, like the rewiring signals that you talked about, or even the decision-related signals where these are related to the response? And it seems like many different brain areas often respond in the same way, such as in your case, you showed FMC and PPC showing similar signals. So I'm wondering, do you think it's even the right question to say, is there the origin of a decision or the origin of the rewiring process going on during the task?
Yeah. So the more one is able to record from large numbers of neurons across multiple brain regions, you are absolutely right, Arun, that one is struck by how much overlap there is between signals in different parts of the brain, particularly in more recent work, just from the last year, in signals related to motor action, or even random actions that go along with the actions for the task that an animal, a monkey or a mouse is doing. And so the issue is, how does the brain make sense of relevant from irrelevant signals, or how does it extract? I'm rephrasing your question. how does the brain extract from all this noise or all this background activity, or it may not be background activity, it is actually related to what the animal is doing or what is going on in a person's head, to extend it to humans. I don't know the answer to this question, but it is more and more clear that there are networks in which brain regions are nodes. So I always refer to the right parietal cortex as a node in an attention network. It is a critical node. I still do not believe that all parts of the brain are equal and do the same thing. That cannot be the way the brain has evolved or works. At least at the lowest levels, clearly, to move your hand, you need a specific signal from a specific part of the spinal cord to a specific set of muscles that make this happen. If you lose or if you learn to move differently, then other inputs may begin to play more or less of a role, and you have plasticity that reflects that learning and hence augments your function. So you need specificity and you need plasticity in order to implement specific actions, yet with learning. How does this principle apply the higher you go? And at the highest levels of, say, of the cerebral cortex, there are multiple nodes, multiple connections. It's not surprising in a way. Even simple analyses of recurrent network, which is a very extreme example of highly connected neurons, you see that all neurons in a recurrent network contain signatures of some input or some task. Yet the output of that network can have specific states. It can amplify small changes in input in order to produce some desired vector or output or trajectory, right? So it is possible that some variation of specificity and plasticity through highly connected networks can create perhaps small world, perhaps specific outputs. In fact, that is the way I believe it must be. So that's a long answer that
no, it's very interesting
window
answer.
into future work.
I think I'll give up my turn to the other.
Thank you, Arun. Thanks a lot for that interesting discussion. Actually, there is one question that I would take up, that's from Gaurav Das, which says, what is the mathematical activation function of human brain corresponding to activation function of artificial neural network? Or I could rephrase it to say, will there ever be one found? Will there be any grand unification theory to describe all the brain function? Will there be any Einstein for the brain function, Einstein theory?
Let me hasten to add that even the most sophisticated artificial neural networks for machine learning, multi-layer networks, do not capture the complexity of human brain connections that neurobiologists understand in terms of the number of neurons, in terms of the number of connections, in terms of the complexity of synapses, and in terms of brain states that are possible. That doesn't mean that you cannot build artificial networks that way, but what is true is that the artificial neural networks that have been engineered with very simple principles do not capture this complexity. For example, the all-to-all connections, the backpropagation that is a way to change network weights. These are all very simple ideas, some of which do not exist in the brain. That doesn't mean that they are not feasible. It doesn't mean that they don't work. But what it does mean is that there are many, many differences between the way we think of artificial neural networks today and the way we know that the human brain works. Having said that, what are the prospects for going beyond present day artificial networks or machine learning to capturing some of the ways in which the human brain and mind do work. And I think there are some interesting possibilities there. I alluded to one or two. The first is that the human brain's uniqueness is that plasticity and processing are embodied in the same element. A neuron processes due to integrating its thousands of synapses and by creating an output that is a nonlinear integration of its inputs. And these synapses themselves are changing. So the processing and the way that a neuron integrates is itself a function of what else is happening. And it does so in a dynamic way. So the hardware of the brain, the neuron and its synapses are very dynamic, that they are changing as a result of the software of learning, if you will. Okay? So that's one difference.
Yeah,
there are
thank you.
Okay,
Yeah.
but I'll give you the example of flying. You know, this is not to say that the only way that an artificial neural network or a computer will carry out cognition is by mimicking the human mind. Not true. Birds fly and airplanes fly. And what is common among them is the principle of flight, that you have to overcome weight by lift and you have to overcome drag by thrust. And birds and planes must follow these principles of physics, but they solve the problem in different ways.
Yeah. Thank you very much. That's interesting take on this. In fact, I do remember reading somewhere that IBM was trying to create chips in which the memory storage and processing could be at the same place to increase the speed of processing and energy consumption. And perhaps we will go towards incorporating principles that we learn from the neuroscience into chips of the future. I would like to invite now Head of the Biochemistry, Professor Subrat Sinha. He is at AIIMS, New Delhi, for his comments. Professor Sinha.
Hello. I actually, at this stage, I don't have anything really to say after this much of wonderful, profound feast, so to say. I don't think I'm coming. Only thing I can say, perhaps, is that sitting in a hospital, I can only hope that with our increasing knowledge of plasticity and the increasing sophistication of human brain mapping, I mean, someday we will find methods and possibly some rules. It's perhaps too much to hope for rules, but well, maybe yes, that help us in surmounting brain injury. And the use of already human brain mapping. I mean, recently there was this article about using those people with deep electrodes implanted in the brain and having surface recordings so as to map the brain much better than that we already do. And using these tools build up from the basics to see whether plasticity and regeneration can help in surmounting the effects of brain injury. So that's something that very recently, with people with strokes because of COVID and all sorts of things, this is something which comes uppermost in mind. That's all. Thank you very much.
Professor Sur, would you like to comment on that?
Well, it is possible that as we understand the multi-connected nature of brain processing, that we will come up with either expected or some perhaps unexpected ways of treating brain injury, loss of function from one brain region by activating or even inactivating other brain regions that are in a very special way related to a region. and so that is actually not an unreasonable idea. And there are many potential therapeutics in play that might be involved. I'm impressed by, for instance, just as a way out example, how deep brain stimulation has become one of the frontline ways of treating Parkinson's. And it's a very invasive procedure, but we have gotten better at doing it. And it is possible that, and the basic idea is that by activating or actually inactivating a node in a complex multi-synaptic pathway and network, you can affect even brain stimulation has been used for major depressive disorder. It has been used for many other kinds of disorders that are essentially neuropsychiatric disorders. And so focal nodes can nonetheless affect higher cognitive functions. And the only way to have a rational view of this is to base it on the networks of the brain that underlie cognition.
Well, thank you very much. And as I said, this is something that we can go on for many hours and we will not be satiated. So it is just amazing, amazing lectures. I must thank Professor Sur for agreeing to deliver this first Foreign Fellows INSA-SERB lecture. It was just a feast for all senses, as I must say, for, his slides, his talk, the delivery, everything. It is just amazing. And thanks a lot for this enlightening talk. I would also like to thank our people who are here on the panel with me, Professor Sinha, Arun Shripati, and Vidita, for interesting discussion and adding to this lecture. And with that, for the last word, I invite INSA President once again, Dr. Chandrama Shaha. And I can see there's a whole lot of people still logged onto YouTube. I can see, but we need to end this. It's at some point in time. Chandrama, you have the last word.
Yes, thank you, Professor Sur, for such an engrossing lecture. And the kind of questions you've got, I mean, there are many questions now that we've not been able to pose to you. And it shows the quality of the lecture and also people getting involved in it. It is really, it has been a feast. I would formally like to thank you for this. And I would also like to thank the participants, Professor Sinha, Dr. Arun, and Vidita, and of course, Dr. Jain for chairing the whole session. And I must thank all the audience who came in today in large numbers. And I'm sure they enjoyed. There have been a lot of appreciations on the net, I can see. And I also would like to thank to Karthik of INSA, who hosted us on this platform. And I have already thanked ACRB for generous funding. Thank you. And with that, we end today's session. And we hope to continue with the series. And really thank you, Professor Sur, for being our first speaker. Thank you.
Thank you. Thank you. It was a pleasure. Okay. I'm signing off now.
Okay, sure.